Welcome. We are here today for a reflection on the history of modeling and theory. This is a podcast series with the Society for Modeling and Theory in Population Biology, which is a new society aiming to provide a forum for discussing theoretical issues and results, strengthening the connection among practitioners in our field, and building a community through meetings, workshops, and online events like this one. In addition, we wish to foster the careers of modelers, help bridge those early days of modeling to become a researcher able to model in population biology. And I really think that this particular series on the reflections of modeling and theory is a wonderful insight into the careers of luminaries in our field who have really a lot to teach us about how to approach models in theory. And here today with me is John Wakeley, and we are so delighted to interview Joe Felsenstein. Joe has influenced our careers innumerably. John and I will probably talk over the next little bit about just how many ways Joe's influenced us. But just to give you a short CV, he's Professor Emeritus at University of Washington in the Department of Genome Sciences and in the Department of Biology. He did his undergraduate at the University of Wisconsin in zoology, his PhD in zoology at the University of Chicago, postdoc at the University of Edinburgh, and then has been at University of Washington as for his entire career as an assistant associate and finally full professor. He's won a large number of awards. I won't list them all, but then they include the Sewell Wright Award of the American Society of Naturalists, the Weldon Memorial Prize, University of Oxford, the Darwin Wallace Medal from the Linnaean Society, as well as fellowship in the National Academy of Sciences and the American Academy of Arts and Sciences, and the list goes on. I should probably stop there. He also won the International Prize for Biology by the Japan Society for the Promotion of Science, which is a really large international award. Congratulations on that. But even cooler, one of the cool things I learned in going over your CV was that a moth was named in your honor. Uphias Felsensteini. Is that pronounced correctly?
I think so.
Okay. That's awesome to have a species named after you. And Joe published a book, Inferring Phylogenies, in 2004. He's also published an online textbook in evolutionary biology that I refer to a lot for a lot of PopGen theory. And his papers have been cited over 150,000 times. And when you go into them and you see the top hits, the ones that have been cited so many times with over 1,000 citations, it's incredible the breadth of those influential papers. Introducing maximum likelihood for tree inference, introducing bootstrap into biology for evaluating confidence in nodes and trees, identifying the Felsenstein zone, which is problematic areas that are almost sure to get the wrong answer in our phylogenetic methods, particularly parsimony. Speciation, love the skepticism towards Santa Rosalia, or why are there so few kinds of animals, should be on everybody's reading list in the field. Recombination, and this is going to be my top one, evolutionary advantage of recombination, where Joe synthesized a really conflicting literature where people had modeled the question of what are the advantages of recombination in such different ways and achieved such different answers. And Joe's paper on this really synthesized and helped us understand what was going on. So another really important paper. In addition, the other thing that's really influential about Joe is he's so nice. He has done a lot of work to build the community, and that is reflected in part, like writing a book and having it online. That's one thing. All of the software development that you have done in order to make it possible for others to apply these models to their data. And that includes Phylip, the phylogeny inference package, which has tens of thousands of users, and Lamarck, a package for likelihood analysis by metropolis algorithms with random coalescence, Lamarck. And of course, you've done that with a team, and many of your students and colleagues over the years have contributed. But thank you, Joe, for all that you've done for our field and for modeling in biology generally. And let's start off with you, John. Questions?
Great. Yes. Thanks very much, Joe, for doing this interview. Your work has been really important for me personally, too, in just learning how to think in terms of trees in biology when I was a graduate student, and very much your work on statistical inference in coalescent theory using MCMC. And one thing that keeps coming up, I go to meetings and a lot of times with mathematical modelers who are more mathematical than either of us. And your paper from 1975, Pain in the Torus paper, keeps being mentioned by people who want to solve that problem still with a variety of means of controlling density in spatial population models in sensible ways. So yeah, thanks again very much for being here. So first question I'd like to hear about, Joe, is when did you first realize that math was a tool that could be used to provide insights into biology?
I think when I was young, I was growing up, I was a very shy kid, and my mother was very proactive about these things. Noticed, I couldn't seem to find any other kids to play with. So she saw me looking at a bird in the nearby park, and she went out and bought me a bird book. In fact, it was Peterson's Field Guide to the Birds. And I got into bird watching, but I was always also good at arithmetic, good at calculation. And there was no particular family background in mathematics. I did have a grandfather who I never met. He died too soon, who was an engineer on one side. But, was interested in math, interested in biology, family interests, a family background of left-wing politics left me with a sense of history, a sense of things weren't the same in the past as they were now, and they wouldn't be the same in the future. And then my high school had a very good English department, and very good history and English teachers, and so that influenced. And I don't know, I always wanted to use the math I knew to help to do something that somebody could find useful. And I remember just trying to figure out ways, and there really wasn't any easy way. And I think it was when I first encountered population biology, particularly population genetics. And that was between the third and fourth years of high school, the junior and senior years. A biology teacher at our high school had connections with the summer biology program in Bar Harbor, Maine, at the Jackson Memorial Laboratories. And I went up there in 1959, which is when I'm 17, and it was still in high school. And I was in the high school program up there. And I did a program surveying mouse populations in the fir forest on Mount Desert Island under the tutelage of a very nice guy, John A. King, who was a behavioral geneticist of mice. And the next year, I came back to the program because the summer between high school and college, and I worked with a behavioral geneticist, John A. Fuller, doing a mouse breeding program at the little mouse room near the mansion where we high school students stayed, and he handed me a copy of a newly produced book. And it was an introduction to quantitative genetics by Douglas Faulkner. And so I started reading it. And that's where, I first came across population genetics. And fortunately, when I was a postdoc, I managed to go to Edinburgh and met Douglas Faulkner. I was able to thank him for writing that book. So I don't know. I think it was first running into population genetics, because all the other ways I tried to use mathematics that would be of any use to anyone weren't.
Oh, that's really interesting.
That's a too long answer.
No, not at all. That's earlier probably than most people in this field in terms of life. Biology is sometimes presented without mathematics, as you know, and oftentimes math is just for math. So it's kind of random points in careers where one sees a connection between the two.
Yeah.
And Faulkner is an excellent, still a classic textbook. A great entry as a high school student. Well, awesome.
It's a very classic book. And people working on quantitative genetics, of course, Mike Lynch and Bruce Walsh have written, two massive and important volumes. But you might pick up Faulkner, and at least if anything is confusing, it could help. Could be a very good supplement to that.
Very much so.
So I don't know. I got into this. I got the sense of, here was a field where they were using mathematics, and I started getting into that.
But your degrees were both in zoology, I'll emphasize, so that the math came early, but your focus still in your degrees was zoology. Along the way, who do you feel really influenced your career, and in particular influenced and encouraged you to model?
Well, I was when I was choosing colleges, and by the way, in those days, I applied to a very typical number of colleges, 4. Nowadays, my son, when he went out to apply to colleges, like 25 colleges, and there are other people applying to a lot more. But I applied to 4 places, and one of them was the University of Wisconsin, and I ended up having an even choice between Wisconsin and the University of Pennsylvania, which was in my hometown of Philadelphia. And University of Wisconsin was 800 miles further from my parents, so that decided it. But I'd heard that they had good biology, and a distant cousin who was contacted through my father, contacted some relatives, and they said, "Oh yeah, Carol Hoppenfels is at University of Wisconsin. Maybe she can help out." And she was very helpful. She went to the Department of Zoology and said, "Here's this brilliant student," and she had never met me. "And here's this brilliant student. He's been in Bar Harbor and so on. Can you help him out?" And they arranged for me to have a mouse room job with a wonderful developmental biologist, Robert Auerbach, who was just a great guy and still is. That was very helpful because I needed the money. But I then, early on, I was in what's called an honors program. It's kind of funny because at the University of Wisconsin in those days, you didn't get honors for getting good grades. You had to sign up for an honors program, which is a rather weird notion. And in that, we had a little seminar, and one time, very soon, James F. Crow, came to give a talk, and I was asked to introduce him, which I did, and I gave him my report from the second year at Bar Harbor, which was quantitative genetics analysis of crossing mouse strains, and he said, "Well, I could come over and hang out at his lab," and it was really the contact with Jim that John Fuller had given me the book, but it was really the contact with Jim. And he also said, you know enough genetics, don't take the elementary genetics course, take my population genetics course. So I did that. And the text material was mimeograph stuff that ultimately became Crow and Kimura's text. So, and Jim was a superb teacher, and was also very accessible to people in his lab. If you had a question about effective population size or something, you could stop him in the hallway and ask it, and he would stand there with his mail in his arms and say, "Oh well, yes, in this case, it's a valid concept, but not in that case." And so hanging out with Jim was really very influential. So he was the first, yeah.
It sounds like that was very early in your undergraduate career if he was advising you about the introductory courses. I think that's unusually early.
I think I started hanging out in his lab in the sort of at the end of my first year, end of freshman year. And the lab, people in the lab included his former student, Moto Kimura, who had come back from Japan for a year to visit again Wisconsin and work with Jim. So Moto Kimura was wandering around the lab, and I got to know his Crow's graduate student Takeo Mariyama, and and of course in the building was Sewell Wright. You sort of see him in the hallways, and there are people like Charles Cotterman, who was the first editor of American Journal of Human Genetics, and he's the guy who co-invented kinship coefficients. Basically, he and Gustav Maleko more or less simultaneously invented the notion.
He's the author of the most famous paper that wasn't ever published.
Cotterman?
Yeah, I mean, his 1940 work didn't really get published besides the thesis until it was in a book much later, yeah. But it was cited and known and influenced the entire field.
Yeah, I mean, what he did was, Sewell Wright had this way of doing inbreeding coefficients that was path coefficients, which worked, but was very hard to understand. It was basically using correlation coefficients and treating alleles as 1 or 0 values. And Charlie Cotterman took it and translated it into probability terms as a coefficient of kinship, and so did Gustav Maleko. Anyway, Charlie was around, Sewell Wright was around. I mean, it was a really great place. I mean, if you're going to train as a population geneticist, that's where you really had to go. And in my third year there, Jim had said, "Would you be interested in staying as my graduate student?" And I thought about it, and I said, "Well, I've been tremendously influenced, but I think I really should try somewhere else. I think I've been influenced as much as I'm going to be here." And Jim was very nice about that. And he said, "Okay." He said, "Well, there's this guy called Richard Lewontin, who is, doing interesting things and making a name for himself. Let me see if I can arrange for you to go work for him for a summer." And he arranged in summer of 1963, when I was between junior and senior years. I went to Rochester, New York, which was where at the University of Rochester was where Dick Lewontin was. And I wrote a program for him for a paper that I was second author on, which was in 1965, which was a simulation of 2x(n) chi-square tables to see. Dick wanted to know how small an expectation could you have and still have the 2x(n) chi-square be okay or a bit conservative. And we answered that question. The number is very small. It's like 1. And later it's turned out Dick had designed the simulation, and later it's turned out that was one of the early validations of a statistical method by computer simulation.
How did you do it?
So
What language and what platform?
the computer there was an IBM 7040, which is a mainframe. It was a decimal, not a binary machine, but a decimal machine made for business. And University of Rochester had bought one of them. And the language was Fortran 2. All my early programming until the mid-70s was done in Fortran, particularly Fortran 2. And in those days, you got one run a day because you punched your programs onto punch cards, took them over to the computer center, put them there was a cabinet that had drawers that were designed for punch cards, and you put it at the back of the submission drawer, and it had in the front of the deck had a card with your user number on it, so they knew who it was. And they'd run it overnight. They'd compile the Fortran program. And when there was data right after that, then they'd run the program on that data. And you had to come back at 11 a.m. the next morning, which is when they guaranteed that the output would be out by. And it was in a rack under your initials. And you pulled it out and you looked through and you said, oh my God, there's a bug. And then you had 1 hour to both scribble on it, and figure out what the correction was. Go into the room that had a bunch of punch card machines. Take your deck, make a correction, punch a few cards. You had to wait, everybody's jostling each other for the punch card machines. Put it in the deck at the right place. Go back outside, put it back in the drawer. And then you got another run the next night. So it took over a month to write the program. And
you could probably do it
longer
on
to
your
run
watch today.
Oh, for sure,
Yeah,
your watch is more powerful.
I was trying to figure out the computers of that era. The University of Wisconsin had a CDC 1604, which was a 48-bit or 32-bit, I can't remember, binary machine. And there they had Fortran also. And I figured out that the instruction cycle on the machine was about 100,000 operations per second. And I was trying to figure out recently, excuse me, I was trying to figure out what machine today has the same speed. And your watch is far faster, and your cell phone is tremendously faster than that. That was the campus machine. I mean, it wasn't my machine, but I finally figured it out. The processor is about the same speed as a singing birthday card. If you get a singing, you open the card and it sings happy birthday to you, and there's a little processor and there's a tiny amount of memory in there and a little loudspeaker, and about 100,000 operations per second. That was the campus mainframe at the University of Wisconsin in 1961 when I first took a programming course.
Funny. I mean, that was a long time ago on the one hand, but also not that long ago, honestly, for that to be cutting edge. And the image of there being people who actually have to run your cards and put them into the computer, do the program, run it for you, and then give you the result. It's so...
The cards were either read in by a card reader, and then there was a magnetic tape machine there, reading these big tapes about this far across, and they put everybody's everybody's decks one after another onto that tape, and then the Fortran run that night, but they'd set the tape up to feed into the actual machine, and it would do compile and run, one program after another. So
that's amazing. it really sounds like you got into research early, and I was wondering kind of a next question to think about. Research goes in fits and starts, and we often spend a lot of time not knowing what's going on and thinking about hard problems and wondering if they can be solved. Do you have a favorite sort of aha moment where you realize something and you can really put the pieces of the puzzle together for you?
Well, you're asking favorite over my whole career or
Yeah,
early
or
on?
any instance that's you can think of.
I think the I can think of a couple that strike me. One, and they both involve using computers. I was interested in computers. I used to read about them in the public library when I was a high school student in the late 50s, and didn't have any access to one until I got to the University of Wisconsin. And when I was in Jim Crow's lab, I would write programs that were genetic simulations, and we're simulating things like truncation selection on multiple loci, and why was it that tighter recombination slowed things down. And I did an undergraduate thesis project with Jim as part of this honors program on modeling two loci with interaction between the loci and recombination. And it was a paper in genetics in 1965. But the interesting thing was I wrote a little program to do, I think it was a haploid two locus model, and these are iterations of a deterministic model. They're not stochastic simulations. And I tried different amounts of epistasis between the two loci. And there was an allele that was favored at each locus. So say the capital A and little a, and the other locus capital B and little b. And capital A was favored, and so was capital B. But then they had different amounts of epistasis between them. And so I was running, and I made them additive on the fitness. And you run them, and they get into a little bit of negative linkage disequilibrium. We knew about D had been around since 1918, but the term linkage disequilibrium had just been coined by Dick Lewontin and Kenichi Kojima in 1960. And I was looking at D and the gene frequencies changing. And then I tried a multiplicative fitnesses. And I started out with D equals 0. And in the additive case, it would then D would go negative. But in the multiplicative case, it just stayed 0. And I looked at that, and it finally hit me that I better do some theory.
Okay.
Because there must be a way, of seeing why in the multiplicative case it was 0. And sure enough, you could do the theory, and that was in the paper. So the algebra in that paper comes from a computer's iteration run, even though I then don't really talk in the paper much about the results of computer runs. But it sort of, once you see a pattern in the computer, you better start thinking about a theoretical explanation for the pattern. So that was one.
For sure.
But another
Yeah.
one was when I was working on trees. I got into that in graduate school with Dick Lewontin in University of Chicago in around 1964 and 5. And there, I started working on phylogeny methods. And when I got to the University of Washington, I continued that as a sort of sideline while I also did theoretical population genetics. And in the mid-70s, I was trying to find conditions under which parsimony methods might misbehave. And I did a derivation. And the derivation gave a condition that was kind of messy looking. And I wasn't very, I thought that's not very elegant. Let me try it again. I may have made a mistake. So I went back through the derivation, did the whole thing over. And I get a different expression, which was also kind of messy. So now I had these two expressions. And at that point, a time-sharing system had come online in the University of Washington's medical school, and I got access to that. And it had a computer algebra system. And so I went and I used the computer algebra system, and it was a matter of having, like I'd done the theory and I had big complicated expression, greater than big complicated expression, or it was product of two expressions, and over here and something else messy over there. And I put them into the algebra system. And I said, this thing minus that thing, and please simplify that. And it came out with an answer that was very nicely, beautifully symmetrical and modestly big.
Sorry, I lost track. Are the two things you're comparing the two different answers you got?
Well, what it was? No, they weren't the two answers. They were the parts, the pieces I had used to get those answers. And the condition was going to be that this thing is greater than that thing.
But incomprehensibly complicated. Was this Maxima that simplified it down or something like that?
I was trying to remember the name of the thing. Is it called Reduce, maybe?
Yeah, yeah.
Basically doing polynomial algebra.
And this was the Felsenstein
system 10.
zone.
Yeah, it was going to become that, yeah. And it was when you were in the zone. So anyway, I put these expressions in, one minus the other, and it came out with something nice. And my immediate reaction was, there's a bug. It's not the same as either of these two messy expressions I got. So I took the pieces on a computer and the answers, the one messy answer I had got, the second messy answer I had got, and the simplified computer algebra one. And I evaluated these messy things to see if they were equal to the numbers you would get from the others. And it turned out that both of mine were wrong and computer algebra was right, and there was no bug. And that kind of, taught me a lesson.
Yeah, there's a few, things there. I think for a lot of people, they see Joe Felsenstein's published papers, and they would never imagine along the way that there were errors and bugs and pulling out your hair and then trying again and fixing your code. But of course, that was part of your path too.
Well, everybody works in different ways, but I basically I try to solve a lot of problems. Some of them problems that people have already published a solution to. I just say, can I get that? Can I get that with a simple model? And it's practice, it gives you a sense of, helps you understand what they did. It's not publishable. And the other thing I do, I just work on a lot of little problems and do a lot of little things. Now, some other people I know don't do any work unless it starts out they start a gigantic project and carry it through. So obviously those are both valid ways of doing things. And I remember once this is another one of those lesson things. During my work on two locus, the effective linkage on directional selection, I had 4 cases. I had a discrete generations model, a continuous generations model, a haploid version and a diploid version of each. So there are 4 cases. And I had this assertion, and I wanted to see if I could prove it. And so I proved it in all 4 cases, doing algebra on, yellow line pads. And I thought, okay, I had to prove scribbled on like one sheet of paper or something, or maybe it was a few pages of a notebook. Anyway, I then lost the notebook, and I said, darn, okay, let me derive them again. And I was quickly able to derive the first 3 cases. And in the 4th case, I tried and I tried and I tried, and I could not rederive it. And I was kicking myself and saying, you lost a pad of paper. Now you'll never be able to figure out that case. And I kept scribbling, and I found a counterexample. I found that it wasn't true in the 4th case. And
thank goodness you lost that pad of paper.
Exactly.
yeah, thank goodness I lost the pad of paper. Otherwise, I would have trusted that and published it. And it also sort of teaches you that if you can't sit down from a cold start without it, your notes, and rederive the whole thing, maybe you're wrong.
Yeah.
So that was
Also,
another
beware
dramatic
of
lesson.
beware of things that just seem to make sense. Also, somehow it makes sense
Yeah.
that you would get the same answer in all of those cases, and it probably didn't bother you when
That's
it
right.
first happened.
And Joe, do you have a favorite paper that you think is underappreciated?
Well, I have this very extreme underappreciated paper. I can give you 2 examples. Let me give you the first one, which it's a method in the early 90s called the bootstrap Monte Carlo method for quickly doing MCMC. And I published it. And using bootstrap sampling and evaluating coalescent likelihoods on the result. And it was really this magic method. And it went out there, and Dick Hudson did some numerical work using it. And he got a hold of us and he said, it's not right. It's overestimating the parameter. It's biased. And we looked into it and discovered that in my derivation of it, I had assumed that a certain quantity was that in sampling a certain way for the Markov chain Monte Carlo, I had assumed I was sampling from a distribution. And it turns out that the quantity that I was sampling, the density function of it has an infinite area under it. It has too fat a tail. And so I had made a mistake. I'd assumed something was a nice distribution, and it wasn't. And it still, to me, is a beautiful, elegant method. And it has only one problem, which is it's wrong. And,
Well, so
it's one of the cleverest things I ever did.
is the basic idea, if I understand right, is the basic idea to get something that you think is proportional to the likelihood by bootstrapping, and then feed it in?
And the problem is that the likelihood curve is not a density.
Right.
Yeah. And it's the impractical part, it would be to normalize it somehow.
Or
I mean, it would be very nice if somebody could figure out how to make the thing work and adjust it somehow to make it work, because it isn't just random sampling, it's sampling driven by you are sampling from the data and making a maximum likelihood estimate based on the sample. And that's one point. And you keep doing that. And you don't have to do nearly as many samples as you would in in a regular bootstrap, in a regular Monte Carlo, Markov chain Monte Carlo. So, okay, but the other case that I want to give you a paper that I particularly feel is underappreciated. In 1978, which is now 46 years ago, I decided to work on, I'd always wanted to do something large and macroevolutionary, and I was inspired by a paper in 1961 where Moto Kimura made the analogy between Haldane's cost of natural selection, the formula for it, and information theory. And he pointed out that these looked connected. And I think that was really a very, very clever intuition on his part. And I sort of tried to work on that area as a graduate student under Dick Lewontin, and I didn't have any results. And then in 1978, I finally got back to it, and I made a very simple model of an evolving ecosystem in which there's energy coming in from a sun, and it's taken up by a whole bunch of organisms, and you weight the organisms by their energy content. And you have a distribution then of the rate at which they lose the energy. And the loss, the hanging on to the energy is both for them and their offspring. You don't actually model reproduction. And then you look at the distribution of these rates of loss and how it changes through time. And you get some very interesting results out in which the population total energy content, under the assumptions I made, goes up linearly with time. And the population gets to contain within it a constant fraction of all the energy that ever flowed in. So, and then you can say some things about how you might describe the biological information. And I was very happy with this, and I published it in American Naturalist in 1978. And it went out there to the world and disappeared. Over the years, I think as of sometime in the 2010 or so, 4 people had cited it. And 2 or 3 of them, it was actually one group. And what they were interested in it was a sort of witty remark I made in the preface, but not the actual model. They didn't care about that. So 3 of the people were citing it for completely uninteresting reasons. And the 4th one was Carl Bergstrom and Michael Lachman, who work on things like energy, information theory in behavioral, they model that. But they don't count because Carl is at the same university. And I had told him about the paper. So the reason he cited it was because I told him. And so that doesn't count. So it basically had no citations. And
that's very funny.
I have recently, I gave a Fisher lecture in 2018, a Fisher lecture in which I went over the model. So maybe that'll get it out a little more. It's getting a little more notice, but not much. I am going to get back to it.
Good. Well, we'll put all of you listeners on alert. This is called, I believe, Macroevolution in a Model Ecosystem, published in 1978 in American Naturalist. So go check it out.
That's right. And cite it.
Do something with it.
And cite it, yeah.
So, Joe, change the topic a little bit. Part of this aim of this series is to get some of your experience and pass it on to the next generations of people. So we're wondering, what for students who are just entering this field, do you have any advice about how to approach theory and the insights it provides?
Yeah, I think you should model things, and model things even if they're not the big problem that you and your advisor have talked about. If you want to learn the field, even go out and find a result in a paper and then say to yourself, I wonder if I could prove that myself. Just I think that what I did as a student and still do, which is to mess about with lots of little problems and see if I can do something, is a good way for a student to, start learning the field. And the other thing is, don't be too realistic. I know people who say, oh, let's make a mathematical model of this. There should be this and there should be that. And then there's predator satiation. And then there's, occasional hurricanes. And, you start putting term after term after term into your model. And you end up with this nice mathematical expression that cannot be analyzed in any way. I mean, you can't prove whether this is bigger than that because there's just too damn many terms, and you don't know what each term ought to really look like. If you try to solve the simplest case that could possibly show the phenomenon that you're working on, if you're working in natural selection, you do start in learning about it with two alleles, a deterministic model, discrete generations. And people might say, oh, that's unrealistic. Don't listen. You want to know how to solve the simple stuff before you get to the complicated stuff. So, and do computer iterations of models. Even if you can't prove what they're going to do, you can watch it in the computer, and if you see something that looks like a generality, then you might wake up and say, oh, wait, can we prove that that's going to be the case? There's also, it's interesting to go back and read some early papers in the field. And early does not mean 1990s. I think papers from the 1900s through 1950s are, it's important to know that they're out there. It's important to know that maybe you aren't the first person ever to think about this topic. I'm always running into people who say, oh, so-and-so did this. And I'll say, no, that was done 30 years earlier by Haldane. And so knowing some about literature is probably a good thing. Download my book. Download my my population genetics book for the population genetics.
Yes, you also make available the bibliography of theoretical population genetics, which is a massive compendium of reference literature.
Yeah, and that was released in 1981, and I never got back to keeping it up. But it's intended to be a complete listing of all theoretical population genetics papers. There are some missing, and there's a few duplicated, but it's almost 8,000 papers at that point.
And I think people don't understand what it meant, because these days you do Google Scholar, but at the time, it was almost impossible to find the thread of a subject, and that just having all of the papers listed let you kind of move back through the literature.
And you can, in the original printed version, I've got a copy here, the big book, there was the listing, and then most of it was concerned with what's called a keyword in context listing, which we had to generate on a computer. A guy named Bruce Taylor worked on it with me. And it just has a line, and for each word, significant word in the title, other than, short common words like and the. So if it's an examination of effective population size in such and such, the word effective is in that listing under E. And so you see all the papers that have effective in them. And it's also listed under population and under size and so on. Now, all you have to do is download the list of papers and put it in any old editor and just search for the word. If you want to find papers on effective population size, you can just search under effective until you'll find all the papers that use the word effective in the title. And it will help you find early papers in the field. So, and when I did it, I actually went to the library and looked up early journals in the 1900s and 1910s to see if I could find unknown population genetics papers that had never been cited. I found very few of them. Almost all of them were known. And the ones I found weren't very important, but I really wanted to make sure it was complete.
Excellent. And I think that's great advice. Tinker, keep things simple, and then look back to the history.
Oh, and compute.
Yes.
Look for patterns in computation. Yeah.
Do you, over your career, do you think people or biologists view modelers and theoreticians differently than when you started? Has that evolved, the role of modeling in biology?
Well, I'm getting old and cranky. You know, you get cranky and say these young people aren't doing it right. Get off my lawn. I've spent all, 49 years before I retired, in teaching theoretical population genetics most years. And it's kind of like, but you sense that biologists are not overly eager to get mathematical. I used to say when I was working on phylogenies that you could clear a room of systematists by using the word logarithm. And that, people go into certain areas because they want to get away from this quantitative stuff. And in other areas, they're a little more open to it in population genetics. I kept having the feeling that I was pushing a stone up the mountain. And unfortunately, I would get it almost up there, and then I'd turn around and the stone would roll back down the mountain. You'd see departments fail to hire anybody theoretical. You'd see departments, hear of labs in your own department where they whispered to their own graduate students that don't bother with that population genetic stuff. You don't have to take that course. Because they were doing cutting edge molecular genetics, and who cares about this butterfly collecting involving populations? Over the years, though, we finally got into a situation in the 1990s where computers and databases, DNA databases, were becoming big. I should say that around 1990, I served for a few years on what was the advisory committee for GenBank. And at the time, we looked back a few years and we said, boy, there were only this many studies in GenBank, sequences in GenBank. But now we have hundreds of thousands. Isn't it amazing? And you could see the curve going up exponentially. And of course, we now look back, and back when I was on that committee, we're way back there at the beginning, but you could see the volume of data was going up very rapidly. And then you started getting major figures in molecular, in what became genomics, molecular genetics that became genomics, suddenly saying to everyone, computers are important. We have to know about computers. We have to know how to do things with computers. And I remember two of them coming through, Leroy Hood, and who later moved to the University of Washington, and David Botstein. And they both came by and they both gave very heavily attended talks to all these people working on molecular sequences. And they all said, computers are important. We have to know how to use biology with computers. It was the beginning of the obsession over computational biology. And everybody metaphorically stood on their chairs and cheered. The audience had said, yes, wow, applause. And I kept looking at them thinking, are these the same people I've been in curriculum meetings with? Are these the same? What happened? You know, why are they suddenly enthusiastic? And there's this great craze for some years and for the phrase. Everybody said, what I'm studying is computational biology. Still, I don't think departments are all that eager to hire a theoretician. I was one of the first people, I think me and Mark Feldman were among the first people hired into a major American department without an experimental lab. I mean, you have to look back and realize that Jim Crow had a big Drosophila lab.
Right.
Dick Lewontin had a Drosophila lab, you know. So you couldn't get hired unless you had an organism. So I think there's still a lot of resistance.
I think it might be a bit better than that. It feels like a lot of departments want a token theoretician for sure, but whether or not they really see it as central in an area to build up is another issue.
Yeah, and that comes up when the question arises: what courses will people take?
Right.
How many? I mean, is it true, Sally, in your department? Does the average graduate student take any theory course?
Yeah, it's about 10% take the
Yeah,
more.
that was similar in the genome sciences department. Well, the genetics department in Seattle, and then what it became, which was the genome sciences department. The percentage is very small. Still
and it could be that you pointed out the 1990s, the big genomics area, that could have been the heyday of pushing people into computational. There's so many programs now that the need to actually be able to program itself may be waning. People aren't focusing on that so much.
Yeah. And I'm not sure. I'm unsure whether we should be pushing programming on the assumption that it will always be important. People are doing a lot with simulation programs now. Now they do have to understand what the model is that's being simulated in the program. They need at least that. So.
And I think what you were saying is you've used your simulations to really give you good ideas, to show the patterns that you're like, I think I could prove something here.
Or to kill off some ideas.
Yeah.
Great. So, Joe, we're getting about toward the end of our time for the interview, and but we wanted to ask you one last question. In your work, you've had a number of really important themes of phylogenetic inference and maximum likelihood in that in particular, impact of recombination, and like I mentioned before, spatially explicit population genetics. These have led to some, small research industries, some of them. What helped you to see the kind of the good problems to work on that ended up being as successful?
Well, I think I was working on, I think I had an interest in population biology a little bit above the species level. I was working on theoretical population genetics. And then I got involved in at first clustering and then phylogenies. And for a long time in the 1970s, John Gillespie, who is the guy, by the way, who first suggested to me in the spatially continuous population that there might be clustering, which I then found a way to prove. And John Gillespie used to say of me that I never worked on the same thing twice. In every paper was on a different topic, which was true. I was trying to find something that would connect with maybe speciation or some phenomenon a little bit above the population size, a population level. But in the 1960s, we had theoretical population genetics, we had diffusion equations, Kimura was doing great things with them. Data was coming along as electrophoretic data. But if you looked outside the species, you said, okay, here's Drosophila melanogaster, over there is Drosophila simulans. How do we think about the difference between them? From population genetics, you got nothing. I mean, you looked and it was a mystery. There must be some way of thinking about multiple species, And I got into phylogenies without thinking that it might even be very important. But by the late 1970s, with more and more people doing phylogenies, mostly by parsimony or distance matrix methods, it hit me that this was one of the ways of addressing multiple species. And only later did I think, well, all those years we were all sort of groping around saying, what's a theoretical framework that we can use for multiple species? Is there some magic model that we haven't seen? And now I look back and I say, the phylogenies. I mean,
the divergence.
That's the genealogy, that's the pedigree of species. And what that has done is to bring the different parts of population biology, the work on multiple species by people like systematists and morphologists, and the work at the genetic level, to bring them again into contact. And coalescence is right. I started working on coalescence because I knew about trees and likelihood in trees, and I started thinking, and then people were beginning to talk about coalescence, and I thought, is there a way of getting this likelihood stuff involved with it? In the mid 80s, I was starting to try to think about methods there. But it's basically
like what you're describing sounds to me like maybe an abiding interest in something particular that you thought was important early on, so things above the level of a population or involving multiple species. And counter to what Gillespie was saying, you worked on that and hammered away at that for long.
I worked on geographic models and I still think actually there is a lot of work to be done on geographic models, because if you ask yourself, we can see things happening in populations and we could do population genetics. We can see, then you look at between species and you've got time scales of millions of years. And it's very, very hard to think about processes working on that scale. How can we think about the connection between them? And thinking about it, the one place that might be important in doing that is looking at geographic differences within species, looking at populations that got separated for quite a while. And I think there's probably going to be a lot more work on that, as the results seem to be relevant to difference between, say, neighboring species. I was working on geographic stuff because of this interest in higher level stuff. I was working on some of the other things I did, not so much the recombination, but the speciation model, obviously. The recombination came out of the early simulation work, which showed in quantitative genetics, gave us hints as to what the effective linkage was. And I was sort of part of that literature. Yeah, I think what appears to me, but wandering around among random papers in the 1970s, is really me groping at some way to get above the population level.
Mhm.
I lost track of your question.
I
Where-
think
how
you
Also,
does-
answered
sorry,
it.
of being shouting at people. I'm one of the generation that thinks that if you're far away, I have to shout into the computer.
No, I think the question was what helped you see good problems, and I think this interview has really helped give insight. And I am so thankful, Joe, for you spending this hour with us and telling us about the influences in your life, how you've kind of groped around with problems, but in the end wanted to understand the diversity of species around us and where that comes from. Thank you.
Thanks.
Okay.
Yes.
I should mention give one credit. You mentioned the package Lamarck.
Yes.
I was involved in that, but the chief mover is Mary Kuhner and her husband John Yamato, who did almost all the work. They did all the coding and most of the thinking about how to use Lamarck. So I shouldn't go letting myself get credit for that. So yeah.
Very fair. And I hope we'll do a transcript and we'll do links to the papers that got called out through this period with you. Thank you so much, Joe.
Well, thank you for the interview. It's given me a chance to rant, which is always good.